Good morning, everyone. I'll leave that for you very quickly. Yeah, ReNeuron, we're a UK-based cell therapy company. Everything we do is allogeneic. We have 65 people in the UK and in Boston. And here's a snapshot of the 3 platforms that we have. I'm going to really only talk about the first one, which is the human retinal progenitor cells today, in the time we have. That is a subretinal delivery cryopreserved cell line that we are now investigating in retinitis pigmentosa. And we have some very good data that I'm going to show you in a second. CTX platform, that's a neural cell. We use that in stroke disability, so chronic stroke disability. It's a pivotal study is ongoing for that. You can forget about it until next year. And if it's positive, you'll notice it will be worth a lot of money. And if it's negative, then we will toss it. Okay? The last one is exosomes. We are harvesting exosomes from our GMP process for CTX. We use these to deliver gene therapies. And we're in collaboration with gene therapy companies to do that. But let's go to the HRPC platform. So first, a little bit on retinitis pigmentosa. It's an inherited degenerative eye disease. There are about 100 different genes involved in RP. So it's not a great place for gene therapy. You have to have a lot of products approved to take care of all of those patients. Luckily, what we do is gene independent. It does not matter what gene defect has caused the problem. There's about 100,000 RP patients in the US. So it's an orphan indication, but it's a very large orphan. The only treatment option is Luxturna in a very small sliver of patients who have RPE65 gene deficiency. All other patients, there is nothing. So HRPC is a compound that we have licensed in from Skepin's Eye Research Institute, which is part of Harvard and Boston. We've been working on that for 10 years now and really progressing into the clinic. We are running a phase 1/2 study. We have completed the phase 1 portion in 12 patients. And we're now doing the phase 2 portion, and I will show you the data from that. In animal models, we could see that we could preserve vision in the RCS rat model, which is FDA's favorite animal model in ophthalmology. We also see that we can that the cells differentiate into functional photoreceptors that are connecting into the optic nerve in animal models. So that gives us great hope, obviously, before we went into humans. We have fast track designation for this and orphan designation as well. Oops, I went the wrong way. So a little bit about the study. So the phase 1 part was a dose escalation from 250,000 cells up to 1 million cells. And we also went from a fresh formulation to a cryopreserved formulation. So it's obviously a little easier to deal with 6 months shelf life rather than 14 hours of shelf life as we had on the fresh formulation. We have 2 sites, Massachusetts Eye and Ear Infirmary in Boston with Jason Commander, and a site in Phoenix with Praveen Dugal. And what you can see on the picture here is actually a picture taken straight off the TV screen in Dr. Dugal's surgery. And it looks a bit blurry because it's actually, they use 3D glasses when they look at that. So you'll have to have 3D glasses to see that clearly. There are 3 ports into the eye, so this is subretinal surgery. It's absolutely bread and butter standard subretinal surgery, but it's still surgery. And these are the results for the first 3 patients in the phase 2 study. You can see, so the way this is done is that, first of all, you treat the worst eye in these patients. You don't want to mess with their good eye. You measure both. So you treat one eye, and then you measure both of them. What you're looking at here is the number of letters on the ETDRS chart. And I put the picture of the chart up there. So that chart is standard, used in all clinical trials. It's got 5 letters on each line, which is why it looks like an upside-down pyramid. Yeah, if you can read, if you can read a single letter, you have 2,800. If you can read 85 letters, you got 20/20, so perfect vision. And then there are degrees in between here. A patient with 35 letters, which is kind of where we're looking at here, that's 2,200. So from 35 to 85 is from 2,200, which is the definition of being legally blind, to 20/20, where you have perfect vision. So each letter is important. We have, on average, a 16-letter gain in a very short period of time in the treated eye, and the non-treated eye is -1. So this is a very strong early signal. Obviously, just 3 patients. One patient, we have 2-month data, and the other 2 patients is only 18 days, so you can see it's going very, very fast. The other interesting thing, you see there's a little dip on the first time point. That's 2 days after the surgery, and that's basically the bleb resolution and the surgery that has a little bit of impact on their vision right away, and then you can see it really kicks in. So this is highly encouraging. Most products that you look at for this, I mean, Luxturna had mid-single-digit VA gains. Eylea will have also single-digit VA gains, obviously in AMD. So, so double digits is really encouraging. So we're looking forward to taking this forward. And with that, I will take your questions.
Well, great. So I guess maybe the first question for you is, how are your retinal progenitor cells different than some of the other initiatives from other companies like Biotime, J-Side, Pfizer?
Yeah, so Asteria, so Biotime now, and Pfizer, they have embryonic RPE cells that are there to support photoreceptors in AMD. I think Pfizer put them in a matrix, while Biotime has a subretinal delivery like we do. So that's different indication, obviously, it's AMD for them. J-Side has similar cell type to us. They're also in RP. The difference is that they use, first of all, they have fresh cells rather than cryopreserved, and the second is that they deliver them intravitreally. And the efficacy results I've seen are kind of single-digit efficacy results, so it's good. It's nice to see that there is efficacy. I don't think they're going to have any replacement, cell replacement or integration, but it will be pure trophic effects. So we're not surprised to see stronger effect than what they have since we go subretinally.
In terms of your cell line or manufacturing of the cells, is there anything you guys do that you can talk about that you think provides for a better product?
Well, we spent 10 years on this. The first few years we had big trouble manufacturing the cells. Finally we had the breakthrough when the innovation was to reduce the oxygen level in the cell culture during manufacturing from 20%, which is ambient air, to 5% in the cell culture. And we have a patent on that, which is the key patent for us.
And you've got the slide here on the early data on these first 3 patients. Can you help us put these improvements you're seeing in the treated eye into maybe a greater context in terms of either other initiatives to treat RP or maybe ultimately what would be an approvable endpoint?
FDA has a guideline on gene therapies in ophthalmology, which is that they define a responder as a 15-letter gain. That doesn't mean that your mean has to be 15 letters. It just means that you need to have a p-value between treated and non-treated. And so let's say if you get a third of patients hitting 15 letters, placebo usually will have 0. You don't need a big study to be successful. Right now we're running at 100%, so if we can keep that up, that's fantastic.
And for the cohort where these patients came out of, how far out does the follow-up go?
This goes for 12 months. It's open label, so we obviously follow the data as they come through. I mean, it is, I think the key question now for us is how long does this efficacy last? We really need a duration of 6 months as a minimum, maybe 12 months. That will be a really strong signal. We like to have a few more patients treated as well. So the next cohort is another 3 patients, and they will be at a higher vision level. So they're going to be between 35 and 60 letters, so between 2200 and 2063 vision levels. In animal studies, we have, when we sacrificed animals, we checked their eyes to see if there are cells still left there at the end of the study. The longest study is a mice study that went to 39 weeks. And there were human retinal cells in the eye, the eye of the mouse, and was a very high survival level at 39 weeks. So that gives us good confidence that this efficacy that we're seeing will be sustained as well.
So what's the timing to begin treating those that next cohort of patients with better vision?
The first patient of that next cohort was treated Thursday last week. So that has just started. There is a lot of, it takes a while because you're very careful here. You don't want to mess with people's vision when they don't have much left. So there's a DSMB stop. So we treated one patient, we wait 1 month, then we do a DSMB meeting, and then we treat the next two. So that cohort will be then finished over the next 3 months or so. And after that, we can do 6 more patients quite rapidly. We don't need to do any more stops then.
And what's the total number of patients in that better vision cohort?
So the phase 2 portion is up to 12 patients. So we will do somewhere between 6 and 12, most probably 12.
Okay, well, great. You know, understanding that it is open label, what should investors look to as maybe the next data reporting from the study?
Yes, so we expect to update the market again on this study in the middle of this year. If efficacy disappears rapidly, we send out the press release right away because that's material. So if you don't see that press release, you don't have to worry. Okay,
and then maybe just I'll ask you one question about the stroke study. As you advance it into the next Pisces 3 study, what were the key learnings from Pisces 2 that are informing the design of Pisces 3?
Yeah, in Pisces 2, we included subjects in 2 different groups. One group were not able to have any movement on their affected side, so basically they couldn't even shrug their shoulder. We did not expect high efficacy in that group, but we included them, mainly because there was a lot of investigator pressure. And then the other group, they had some movement on the affected side. The outcome of the study on modified ranking, which is what FDA wants to see, which is the only endpoint where anything has been approved, which is TPA from Genentech and the acute stroke. We had 35% efficacy in the overall patient group, and we had 50% efficacy in people who are able to shrug their shoulder. And the difference is if you can't shrug your shoulder on the affected side, that probably means that the cortical spinal tract is not intact. So that means even if you're giving some good efficacy into the brain, it's not going to translate into any improved movement. So our Pisces 3 study, which is ongoing, has 110 patients in the US, 1-to-1 randomization between CTX cells, which is 20 million neural stem cells injected into the basal ganglia on the affected side in stroke patients 6 to 12 months post-stroke. They need to be moderate to severe disabled. And the placebo group is sham surgery. So they are actually sedated fully. The surgeon will make an incision, but nothing is injected. The patient will not know if they received treatment or not. The surgeon will not be blinded, but the assessment centers, they are blinded.
Okay, and has the company given a projected timeline to either complete enrollment or report out top line?
Yeah, so we're reporting data on that next year. And since the phase 2 study was 23 patients, we had very strong results, but it was a single-arm study, so it's very hard for investors to interpret that. So I think there's very high expectation to see control data in that indication. And there's obviously nothing available for disabled stroke patients. These are patients that are kind of in a wheelchair or using a Zimmer frame at home and need help at home to live a normal life. And if we can make a huge impact to their life, that would be a big deal.
Okay, well, we'll look forward to the company reporting data from both of those programs. Thank you.
Thank you very much.